Okay, I think that people can hear me, but I guess that Faith introduced the subject of today, and hopefully I will be able to share with you some of the data that we have regarding the non-specific immunological effects that are induced by BCG vaccination, and how that can be used, hopefully, in the fight against the new coronavirus. So, if I could have maybe the slides. Yes, thank you. What I would like to share with you is the data regarding the BCG-induced trained immunity and how can this be used as a tool against COVID-19. If we think of COVID-19 and how it spread from a new coronavirus infection that has occurred for the first time at the end of last year to become now a pandemic. Now we have millions of people being infected. We have, unfortunately, hundreds of thousands of people died. And also, these had huge consequences for the healthcare, for the society, for the economy, for the economies of all the countries around the world. And this is just a picture of what has happened in one of the days in the last week or so, in which a lot of confirmed cases and deaths have been described. Now, the final response to against COVID-19 in the end will be a vaccine. And the race for vaccines has started with more, if I'm not mistaken, more than 80 different groups, either academic and in industry, trying to develop a vaccine. But the problem is that being a new infection, there will be time needed to be able to do all the research needed, to do the preclinical and clinical studies, and also to start manufacturing and get the approvals to use the vaccine. Even in the most good scenarios, we are still around 1 year, if not 1.5 years, from the possibility to have the vaccine on the market for all that would need it. So until now, we would need some other ways in which to treat the disease, and eventually, if possible, to have approaches to prevent the disease. Now, of course, in terms of treating the disease, antiviral therapy is being tried. There are several trials which are trying to identify which is the best immunotherapy available, either from type 1 interference to anti-cytokine therapy, because a cytokine storm has been described to be part of the severe cases that end up on the intensive care. But the question is, would it be also manners in which we can improve in which we can prevent the infection? And in these thoughts, one of the possible answers is BCG vaccination. And why BCG vaccination? When BCG was introduced in Sweden for the first time almost 100 years ago, people have described that the mortality due to BCG is actually that mortality due to infections in children from 0 to 4 years is very significantly decreasing from 11% to 4%. And that was not due to the TB deaths, because TB deaths were responsible for less than 1% of the deaths. This is, of course, something that happened before the antibiotic therapies, when a lot of children, unfortunately, died because of infections. So people have observed this epidemiological effect of BCG many times during the coming years. And this is actually a report of WHO assessing various trials in which people have looked at general mortality in the children who required and received BCG vaccination. And you can observe that in all these trials, and you don't need to look at all the names and studies, because the slide is very small, but all the mortalities are on the left of the line, which is actually meaning that BCG had a protective effect. And the conclusion of the WHO is that BCG is likely protecting against all-cause mortality, and this has been shown to be due to respiratory tract infections and to neonatal sepsis, and that more studies need to be done to understand exactly what is the meaning of that and how can this property of BCG be used. In addition to all these studies in children, there are also 3 additional studies that have been done in adults or in the elderly. There is the study of Nemes and colleagues, which was a revaccination with BCG for TB, but in which they looked also at side effects, including respiratory tract infections, and they have observed that BCG reduced respiratory tract infections by 73%. There are also thereafter 2 rather small, we must say, studies, one done in Indonesia and one done in Japan, which both of them have shown that vaccination or revaccination with BCG results in less respiratory tract infections. Now, how is possible actually that this is happening? Because one vaccine in principle should protect against the target disease, which in our case is tuberculosis, whereas here we see the reduction in more than one type of infection. Now, if we look at how is the host defense taking place, we always in the last half a century or so divided the host defense in either innate and or adaptive. And innate immunity is what is happening in the first days or hours basically after an infection. Myeloid cells, neutrophils, macrophages, and so on accumulate at the site of the infection. There is phagocytosis, elimination of the microorganisms. These are all very rapid and effective immune responses, but they tend to be non-specific and indiscriminate, and it has been always hypothesized to lack immunological memory. On the other hand, if the infection progresses, the minority of the infection which is not eliminated, then T and B cells come into place, adaptive immunity is built, and there is specific activation against a particular microorganisms through antigen presentation and recognition, and that also builds long-term immunological memory. The interesting thing is, however, if we look at immunological memory as the ability of a system to store and recall information, and this is evolutionarily speaking very advantageous. So it would be very strange to see that actually only 5% of all organisms on Earth and all species, much less organisms, but all species on Earth have adaptive immune memory, because all the others, insects, plants, cephalopods, and so on, they actually, mollusks, annelids, and so on, all the invertebrate animals and all the plants, they do not have adaptive immunity. They have only innate immunity. So it would be very strange then immunological memory, which is very advantageous for the survival of a species, would have evolved only in 5% of all species. But the question is, of course, is that actually true? And if we look in the literature at the plant immunology literature or immunology in insects or sponges and corals and so on, we observe many instances in which it has been described that innate immune responses in these different types of organisms, multicellular organisms, do adapt, do build a certain memory which can protect them later on during an infection. And the same has been described actually also in case of vertebrates, all studies showing that SCID mice, severe combined immunodeficiency mice, in certain situations, for example, vaccination with BCG or mild infections with either Candida or murine CMV virus, they can develop protection later on against an infection through a T and B cell independent mechanism. Now, we wanted to see whether we can reproduce this old data, and indeed, this is a study that we have done 8 years ago, in which you observe that vaccination of SCID mice with BCG can protect against a lethal Candida albicans infection, a disseminated candidiasis model. And you see these are the non-vaccinated animals and the vaccinated animals, which are completely protected. And this is associated also with less fungal burden. Now, how is that explained? And people have done many experiments, and due to time, I'm not going into detail, but what is happening is the reprogramming of the functional capacity of the innate immune cells, such as monocytes, macrophages, and K cells, to respond better to a stimulus. And how is that mediated at molecular level? That is mediated by an epigenetic reprogramming at the level of the myeloid cells. A myeloid cell, let's say a macrophage in resting, it has very dense chromatin. It is very densely packaged together. It's like a closed book. Whereas during an activation, during an infection, and so on, there are chemical modifications in the histones which open the chromatin, and the information contained in the DNA can be read to lead to transcriptions of genes which are important for host defense. This is an open book, basically. Thereafter, a lot of these epigenetic changes are lost, and previously we thought that everything returns back to normal, but it is not completely the same normal, because some chemical modification, especially monomethylation of histones in certain places, especially the lysine 4 on histone 3, remains methylated, and this serves like a sort of bookmark. Although these chemical modifications are not enough to induce gene transcription, they still mark the genes which are necessary to be stimulated and transcribed next time that we will see an infection. And when that is happening, we can open the book, let's say, we can open the chromatin, we can induce gene transcription much quicker and much more efficient. And that is what is happening during these processes. And this is how this hyper-responsiveness, this adaptation of innate immune cells take place. This leads actually then to an improved secondary response to a secondary infection, and that leads to protection. This has been described also actually in tolerance, where the opposite effect is taking place, where tolerance is induced, for example, by very severe sepsis. But in case of BCG vaccination and in case of certain mild infections, training of the innate immune response takes place, and an improved responsiveness is induced. Now, we were interested to know which are the genes which are important to open, in which the chromatin is more accessible after such an infection with either BCG or with Candida albicans, for example, with beta-glucan component of the cell wall of Candida albicans. And we have done a genome-wide assessment of important marks of open chromatin, H3K4 monomethylation, trimethylation, and H3K27 acetylation. And what we have observed is that a lot of genes important for signaling, for innate immune inflammation, for innate immune activation, such as toll-like receptors, interleukins, G protein signals, and so on, integrins, they were more open. They were marked at epigenetic level after a first vaccination or infection. In addition, we have observed also important changes at the level of metabolism. And why is that needed? And again, due to time, I'm not able to go through all the experiments, but go directly to the conclusions of what it has been observed. There is a complementarity between the innate immune signaling through pattern recognition receptors, metabolic rewiring in the cell, and epigenetic reprogramming with increased function of the cells. How is this taking place? For example, in case of the BCG, there is interaction with pattern recognition receptors. In case of the BCG, the NOD2 recognition of peptidoglycan is the most important, which leads to a stimulation of the pathway starting from PI3 kinase ACT mTOR with activation of HIF1 transcription factor, which thereafter, as a first effect, strongly activates glycolysis, and the energy metabolism shifts from oxidative phosphorylation to glycolysis. In addition to that, there is replenishment of the Krebs cycle from glutamine with glutamate, alpha-ketoglutarate, and accumulation of fumarate. And why is this taking place? Because fumarate, which accumulates in these cells, is an inhibitor of KDM5 histone demethylases. And when these histone demethylases are inhibited, then the methylation of lysine 4 at histone 3 is allowed to stay put. That means that the chromatin can be marked at the places that are needed. At the same time, there is also upregulation in the cholesterol and mevalonate synthesis pathway, and mevalonate provides an amplification loop of these processes through the IGF-1 receptor. All this combination of intracellular immune signaling with metabolic changes and epigenetic processes lead to the fact that these cells are more active upon restimulation. But is this what is also happening actually after BCG vaccination? And this is a proof of principle trial. And in the proof of principle, in this first proof of principle trial, we have now vaccinated healthy adults. We have gone to healthy young adults who kindly donated blood before and after BCG vaccination, and we assess the capacity of their immune cells to stimulate cytokine production capacity if they are exposed to either Mycobacterium tuberculosis as a specific stimulus or to Staphylococcus aureus or Candida albicans, which are completely non-specific, obviously. And what we have observed is that the production of interferon gamma, for example, was strongly increased not only after stimulation with a specific stimulus, but also after stimulation with Staphylococcus or Candida. This was happening not only at interferon gamma level, which is produced mainly by lymphoid cells, either NK cells or CD4 cells, but also at the level of the monocytes, which produce TNF or IL-1 beta. And although the changes are not that strong as with interferon gamma, you can observe here that also the non-specific capacity of the monocytes to respond with production of TNF and IL-1 beta is increased by the BCG activation. As BCG vaccination, as shown previously in vitro, we have also isolated monocytes before and after BCG vaccination, and we have observed also strong changes in the epigenome of the monocytes at the level of histone modifications. This is the case for H3K27 acetylation dynamic regions, which are strongly increased in the responders. And there are several pathways which are strongly upregulated, and I just want to point out again NOD2, which is the receptor for BCG. Now, these in vitro and proof of principle studies, are they also accompanied by a true protection against an infection? Of course, now it is difficult to infect our healthy volunteers, but what we can do is to revaccinate them later, 1 month later, for example, with a yellow fever vaccine. And why did we do yellow fever vaccine? Because this is an attenuated virus, and we actually induce a very mild infection in these volunteers. We can follow with a PCR the virus in the circulation. It's just the pure viremia, which is eliminated within approximately 1 week. So yellow fever vaccine viremia, I must say, was very low on day 3 because it was just starting to go up. And it was also very low on day 7 because we eliminate the virus, which should happen. But on day 5, when the maximum of viremia is present, we see a very significant downregulation of the viremia in the individuals who were vaccinated 1 month before with BCG. Now, very important is the fact that despite this lower viremia, the yellow fever antibody titer, which you can observe here in the right upper corner, is completely normal. So despite the lower numbers of viruses, antigen presentation was very effective, and the antibody titers were absolutely normal, even if individuals got BCG and the viremia was lower. Interestingly enough, when we tried to identify the correlate of protection, we were not able to identify interferon as a correlate of protection. We tried both type 1 and type 2 interferons, but what we have observed as an interesting correlate of protection was production of IL-1 beta and IL-6, not of TNF pathway, but the increase of the monocytes in their release of IL-6 and IL-1 beta was strongly correlated with the decrease in the virus. This is, of course, the CT value, and the CT value is inversely correlated with the number of viruses, but you can observe here, the higher the IL-1 beta upregulation after BCG vaccination, the lower the virus. Now, all is fine, and I showed you changes that take place 2 weeks after vaccination and 3 months after vaccination in the monocytes in the circulation, but that is in itself kind of strange, because monocytes in the circulation are there only for a couple of days. They are continuously replenished from the bone marrow. So how is it possible that we observe 3 months later, for example, and we have seen some differences also after 1 year later, if those monocytes are no longer there? So the only possible explanation is if similar type of changes take place also at the level of bone marrow. And work of Mas Divangahe has shown that this is happening in the mice, but we wanted also to know whether this is happening in humans as well. And in a follow-up clinical trial, we asked the volunteers who got vaccinated to donate also some bone marrow aspirate before and 3 months after BCG vaccination. And this is exactly what was happened. 15 healthy volunteers donated blood before and after BCG, and we had also a non-vaccinated group as a control group. The immunology was the same, but the very important was when we purify, we isolated the hematopoietic stem cells, and then we performed on this isolated cell population RNA sequencing. We have seen very strong changes in the bone marrow hematopoietic stem cells 3 months after the BCG vaccination. And as you can observe here, when we performed pathway analysis of the changes taking place in the stem cells, there was a very strong bias towards myelopoiesis and granulopoiesis. We have observed similar changes also if we look at the level of ATAC-seq. So chromatin accessibility was also changed, just as the transcriptional programs. So it seems that BCG vaccination induces changes at the level of bone marrow, myeloid cell progenitors, with a strong bias, and we see now also improved responsiveness of these myeloid cells in the circulation. So to conclude, BCG vaccination can lead to non-specific protection against unrelated infections. I showed you some data on yellow fever viremia, but we published last year also a study in experimental human malaria. It's inducing what we call trained immunity, upregulation of the function of myeloid cells in circulating monocytes, but also it's reprogramming the myeloid cell progenitors in the bone marrow. And it is also responsible, but also this is in TB, and I didn't have time to show that, for the early clearance of TB in healthy volunteers. Now let's go back to COVID-19. So COVID-19 is a severe, it's a viral infection which can vary between being very mild in the majority of the individuals, actually being mild, towards very severe, and unfortunately in some individuals leading also to death in other individuals. And there are several major risk factor groups, especially the age, but also people with comorbidities, people with diabetes, cardiovascular diseases, and so on. So what SARS-CoV-2 virus is doing, it's recognized by ACE2 in the epithelial cells which are invaded, and thereafter it's inducing also immunopathology at the level of the lung. And the immunopathology is due also to release of danger signaling mechanisms and accumulation of a lot of myeloid cells, neutrophils, macrophages, and so on at the place of the infection, which can, on the one hand, can contribute to elimination of the virus, but they can induce also collateral damage. Now, why would we think that BCG activation could protect? Because BCG vaccination, it's increasing these innate immune responses. Would it be possibly also dangerous because it can improve the inflammatory response? Well, we do not think that that is the case, and for the following reason. We have this BCG, first of all, has been used for a long time, and similar type of lung pathology due to hyperinflammation is also happening during influenza infection. And in influenza infection, we know that BCG is not increasing it, but rather decreasing the mortality there as well in experimental models. Secondly, what we have observed also in the yellow fever vaccine study is that a high innate immune response, which was exemplified by the high IL-1, IL-6 production capacity, is able in the very first phases of the infection to decrease the viremia because the innate immune responses are more effective. This low viremia can lead to low systemic inflammation, low symptoms, and survival. whereas we believe that low innate immune responses, which can permit the virus to multiply, can lead to viremia with a late surge in inflammation, which can be deleterious, which can lead to high severity and death. And in this case, of course, blocking the autoinflammatory loop, for example, with anti-IL-1 or anti-IL-6 therapy, as it is currently tried in patients on the intensive care, could prove beneficial. Similar type of processes have been described, for example, if we are to make a comparison in meningococcus sepsis. Precisely the same is happening in meningococcus sepsis, and it has been shown that children with high production capacity, paradoxically, children with high production capacity of cytokines in the beginning, they have higher chances of survival because they have lower inflammation and lower symptoms later during meningococcus sepsis. That being said, the hypothesis has been made by several groups, and we have started that as well, that BCG vaccination might have a role for non-specific protection against COVID-19. Now, there have been also, in the last month or so, several ecological studies showing that countries which are associated with BCG immunization programs have lower number of COVID-19 cases and also lower mortality. This is one of the examples of Miller. I think this was the first study, which was, I think, end of March or so, published on medRxiv, showing that countries without a BCG vaccination program have higher mortality compared with countries with BCG vaccination program, and this has been now reported, this type of analysis, by several other groups. One of the most, let's say, compelling arguments has been made in Germany, where East Germany, which was communist until 1989, had a BCG vaccination program compared with Western Germany, which did not have it. And both morbidity and mortality are lower in the east of the country. However, we have to be very cautious considering this type of ecological studies, because many differences can be present between countries, different in the genetics of the population, differences in the way that the epidemics has evolved in the different countries, differences in demographics. For example, a lot of BCG vaccinating countries are developing countries which have a much better demographics in terms of COVID-19, with much higher proportion of young people. There is also differences in which the COVID-19 is diagnosed and reported and so on. So it is very different. Some of these challenges have been shown also by studies which was published actually 3 days ago in Journal of American Medical Association. And I think in terms of epidemiological studies done now on COVID-19 and BCG, this is probably the strongest. Israel had a BCG vaccination program until 1979 or something like that. And colleagues from Israel have analyzed COVID-19 in the individuals in the last 3 years of vaccination and compare that with the individuals in the first 3 years of that vaccination. And in these individuals, these two important cohorts in the same country, having the same type of environment, let's say, they were not able to see differences. However, this was only done in terms of approved COVID-19 diagnostics, but it said nothing about the severity of the disease because they didn't have that information. It was purely from the database. The fact that a BCG at birth is unlikely to give several decades later a strong protective effect is somewhat suggested also by an older study that we have done, in which we have shown that after 1 year of BCG vaccination, the heterologous effects are still present but start to wane. So, and this is also coupled with epidemiological studies in the children showing that the heterologous protective effects are mostly staying for 3-4 years after the BCG vaccination. That means that the only way in which we can and should decide whether BCG is useful for the fight against COVID-19 is to perform new randomized prospective clinical trials in which people are randomized to receive either a placebo or a BCG vaccination and then follow up the disease. This is ACTIVATE study, which actually has been started by us together with the colleagues from the University of Athens, the group of Evangelos Jamarelos, already 2 years ago, because we wanted to perform this study and assess the capacity of BCG to protect the elderly against infections even before the COVID-19, because we wanted to know whether a protection due to influenza, seasonal influenza or other seasonal infections could be present. So we started this 2 years ago, and we are actually very close, some 1-2 months before knowing whether in this small study in which elderly were vaccinated either with BCG or with placebo, we will know whether they have more or less infections. These are elderly individuals who were hospitalized for different reasons, and at the moment that they went home, they were randomized to receive either BCG and placebo. And in this group, we already know that their susceptibility to infection is very high. So hopefully we will see a difference in infections between these groups, and hopefully, of course, BCG would be protective. There are several other clinical trials that have been started. We have started another study in healthcare personnel because they are in the first line and have high risk of contacting the disease, the COVID-19. And we have already recruited 1,500 volunteers working in 7 hospitals in the Netherlands. We included them from 15 March to 20th of April, so almost now, 3 weeks ago or so. And we are following them, whether they get sick, whether they get the infection or not. And every 2 weeks, we perform an interim analysis, and we will stop the trial at the moment we see beneficial effects. And at that time, everybody will get the vaccine. And more or less the same will happen in another study that we have also just completed a couple of days ago, in which 2,000 elderly individuals above age of 60, they were randomized to receive either BCG vaccine or placebo. And we are also following them up now to see whether we can observe a protection due to BCG. There are other clinical trials ongoing in addition to those in the Netherlands. There is the BRACE study, which has been started in Australia. We'll continue, we'll collaborate also with the colleagues from the group of Nigel Curtis to collect a total of 10,000 healthcare personnel, so to have even more power to observe something, an effect of BCG. And started or under preparations are BCG studies in more than 7 or 8 countries. I think there are 10 or 15 trials already being prepared with BCG. In addition, there are also trials being prepared and started in Germany and India with VPM 1002, which is modified BCG, which contains listeriolysin gene, which allows the BCG to escape the lysosome. And this has been shown in preclinical trials to be even more effective as BCG, and hopefully that could have also an effect. And there is another vaccine which has specific beneficial effects, oral polio vaccine, and it is considered now in a large clinical study at the NIH in United States. So all in all, what we hope to achieve is to use BCG or other vaccines which induce strain immunity to boost the capacity of the innate immune response to handle the virus, to decrease the viremia, and then thus decrease the symptoms and the severity of the disease. And this, we hope, can be a tool actually that can be used against COVID-19, but also against other infections in the future, until the development of and use of a specific vaccine is available. For a new vaccine, we would need 1.5 to 2 years. And in the meantime, we hopefully may use trained immunity-induced vaccine to induce a boosting of the capacity of the immune system of the population to respond to a new infection and to bridge the period until a final good solution is available in the form of a vaccine. And finally, of course, I would like to thank the people from our group who are working on the project, the colleagues from Utrecht and Athens and Melbourne with whom we are collaborating in the BCG vaccination trials. Thank you very much. I'm not sure actually if, okay. Now finally we hear you. We were not able to hear you during the introduction, but doesn't matter.
That's very disappointing. But I hope I'm checking now with the audience if someone can hear me, because it will be important to transmit the questions. Thanks for a great seminar. Thanks for a very great seminar. I just checked if they can hear. Can you hear me? Could someone type?
I can. Yes. I think everyone can hear you. Yes.
Okay. Fantastic. So we can start. There's really a lot of questions, but the first ones were: does the reprogramming only occur in myeloid cells or also mesenchymal stromal cells, because they also express pathogen recognition receptors? And then just hold on to that. There's a general theme about memory, and because you're looking at immune cells that have a short lifespan, how can we think about this in the context of memory over years in life? So if we can take those two, and then I'll bring the next up.
Yes. So actually, this adaptation of the function of the cells through epigenetic reprogramming is a very old mechanism. It's present, is described in plants, it's described in invertebrates and so on. So this is a very ancient mechanism, and it will be very strange to happen only in immune cells. And actually, it has been shown in beautiful studies to happen also in epithelial cells and endothelial cells. It's a very old mechanisms of adaptation of cells. That being said, I do think that the immune cells in general, the myeloid cells, but also the NK cells, it has been shown also in NK cells, they are more efficient in building up this adaptation. I do think very few studies, I don't know if any study has been done in mesenchymal cells, but I do think that changes will happen there as well. So I do think, although by far not enough studies have been done, I think that at that level, there will be also very important findings to be made and discoveries to be made, because I think that is happening there as well. So that is the first part of the question. The second part of the question, how can we consider this a memory, a true memory? Well, it is not the memory of the cell. It's obviously it will not be the same cell. We do not have a memory monocyte, for example, or a memory macrophage as we have a memory T or B cell. So the changes, the epigenetic changes take place in their progenitors in the bone marrow, and that's where it happens. So it's more, let's say, a memory of the entire innate immune lineage, entire innate immune response that is taking place, not a memory of a cell. And according to our data that we have, this is still efficient for maybe 1, 2, maximum of 3 years, I think. I don't think that, for example, a BCG at birth is able to be very efficient a couple of decades later. These epigenetic changes, because they happen in progenitor cells, they can be also lost. And they are lost as we have seen in our studies. After 1.5 years, they started to be lost, and that's why actually induction of trained immunity also against COVID cannot represent a long-term response in terms of, well, you do vaccinate once with BCG and everything is fine. This is really a bridge until we have a specific vaccine which can induce protection for maybe, hopefully, 20, 30 years.
Great, thanks. So then there's one question about whether latent TB could also be protective. And next to that, there's an increase in IL-6 that's caused by BCG vaccination. And patients in ICU have been reported to have cytokine storms because of IL-6. So could it be harmful?
Yes. So the first one, what was the first story?
Latent TB.
Latent TB,
could
yes.
It also be
latent TB.
Yes.
We do not know, actually. There are no studies done, but on the immunological level, there is a beautiful paper in Journal of Clinical Investigation published, I think last year or the year before, 1 or 2 years ago, by the group of Tom Ottenhoff, showing that latent TB patients have also an improved capacity of their monocytes and macrophages to respond to tuberculosis, to Mycobacterium tuberculosis. Actually, their cells were even better in their trained immunity phenotype. That being said, we don't know further because nobody has looked at the clinical endpoints, but purely at the function of the myeloid cells, they are increased also in individuals with latent TB. Further than that, more studies needs to be done. Now, regarding of the cytokine storm, which is very important, and actually this is the aspect that we were also asking ourselves before starting all these studies. Would it be dangerous actually to induce trained immunity in these individuals? And as I mentioned also during my presentation, we believe not, for several reasons. The first and most important reason is this should have been observed basically because BCG was given also during influenza vaccination, during influenza seasonal epidemics. And if that would have been the case, we would have had epidemiological studies showing that BCG aggravates some of the patients with influenza, because influenza people who die of pneumonitis on the intensive care also have the cytokine storm. It's not precisely the same, but is very similar to COVID-19. That is one argument. The second argument is the studies that we have done with the yellow fever vaccine, in which we observe if we improve the response of innate immune cells in the beginning, at the time that the people get the infection, then you actually decrease the viral particles, the viral load, and that leads to decrease inflammation during the infection and decrease symptoms. And the final argument that I didn't have time to mention is the fact that also we were wanted to be very careful. So we asked in the Activate trial, which was already going on in Greece because the people were already vaccinated, we asked the permission of the ethical committee to allow us to do a safety interim analysis after 6 weeks in the COVID-19 pandemics. And that has been done, and there was no safety problem in the trial. So because of that, we are now confident that we are not doing harm. We don't know if we will protect, but at least we are not doing harm. Okay.
So next batch of questions. The route of vaccination for BCG, does it matter? Intravenous, intradermal? The strain that you're using, someone says the BCG vaccine is very heterogeneous. So what strain are we referring to? And
yes.
Then a final one is the timing of, so you detected programming 3 months after vaccination, and why you're expecting that residual inflammation that late. Does that make sense? I hope I've captured the question.
So if I'm starting with the last one, we do not have inflammation in the circulation. We have increased responsiveness of the innate immune cells to the re-stimulation. And this increased capacity to respond is due to the fact that the bone marrow progenitors have been changed. So the transcriptional program of the bone marrow progenitors the hematopoietic stem cells, and the MPPs, actually the myeloid progenitors, have really changed, and they produce continuously daughter cells which are modified transcriptional program, which are able better to respond to restimulation. This is regarding the long-term effect, is because of these changes at the level of bone marrow progenitors. Regarding the first questions, let me think.
That was the strain and then the route of administration, yeah.
Yes, the route and the strain. So regarding the route, at this moment, BCG is given intradermally, but I cannot exclude that another route would be more effective. But of course, this is not in use now in humans. But for example, there was beautiful work being done and published beginning of this year with the intravenous route of administration, at least for TB, and showing stronger effects in non-human primates if the BCG was given IV. Similar studies have been available also in the mice. So maybe this effect of BCG would be even stronger IV, but of course, in terms of possibility to give IV to a large population, that would be much more restrictive. So at this moment, we are just trying to do the standard route of administration. Regarding the strain, this is a very important point, and of course, at this moment, there are several important strains of BCG in the world. There is BCG Russia, which is the most used, is BCG Denmark, it is BCG Japan, and it has been reported that BCG Japan especially, it's more effective, more immunogenic than Denmark and Russia, and probably Russia is a little bit less than the other two. Our studies are being done now with BCG Denmark because that is the BCG that is available in the Netherlands. But I could envisage differences depending on the strain of BCG. They have been reported, and there could be differences if these studies are being done with different strains.
Okay, so another question is, in terms of training immunity, is BCG the best model? Could other bacteria do it, other parasites? Yeah, let's go for that.
So BCG is definitely not the only one. We started with it because we worked with it and it was the most studied. And the strongest and the largest number of studies are done with BCG. But there are at least 3 vaccines which have shown also protective non-specific effects. Is oral polio vaccine, which I mentioned, is also considered now in the United States. Another one is measles containing vaccines, which have been reported, especially in large epidemiological studies in Africa, that can be protected against mortality. And another one is variola vaccination. This is not available anymore, of course, but there are studies showing that people who have received some time ago variola vaccines are protected against other types of infections. But from all these, BCG, measles, and OPV are good candidates, and I would not be surprised to be others. BCG is also not ideal. We are not in the middle, but we just completed a study in 300 healthy volunteers to look at the immunological effects of BCG, and we observed that proper response to BCG is only in approximately 50% of individuals, at least here in Europe. We don't know yet in other countries, but in European population. And we have seen that also in our protection studies against yellow fever vaccine, viremia, or against malaria experimental infection. BCG is not ideal. It's inducing potent strain immunity only in approximately 50% of individuals. And we are looking now at ways of improving that. One of that is the VPM 1002, which was developed by the group of Stefan Kaufmann at the Max Planck in Berlin, which is now also in clinical trials. But there are probably also other ways of improving the effect.
Okay, so another question that's coming through is ethnicity and whether we're seeing protection in some groups compared to others. So from India to Brazil, everyone's asking about their country.
Of course.
Yes. So whether you could share any info on that.
So There have never been a study, as far as I know, really investigating at the same time with the same protocol and everything, different people with different genetic backgrounds and ethnic backgrounds. So we don't know that for sure. But in terms of non-specific beneficial effects, they have been reported basically everywhere. I'm sure actually that there will be some differences between population because of the environment, and of course on TB, for example, on the effects on TB is very well known. It's a very well-known difference in terms of latitude. The northern latitudes appear to respond better to BCG in terms of protection against tuberculosis than the lower latitudes. And nobody knows exactly why and what is the cause. Some people say it's probably because of the non-tuberculous mycobacteria in the environment, possibly also genetic background of the different populations, but we don't know actually precisely. In terms of the non-specific effects of BCG, we don't know exactly whether this latitude difference is also present, but non-specific beneficial effects have been reported everywhere, including Africa, South America, Europe, North America. So I suspect that a level of protection, if there is, it is given in all the population. And maybe there are some differences, but I think that in all the population there will be protective effects.
Okay, there's another one here that you've said that you proved that BCG vaccination leaves epigenetic memory in HSPCs, the paper by Shirovic. Could you please comment on the fact that you don't see lifelong protection? Is it because you think the affected cells are progenitors instead of long-term HSCs? And do you think other types of vaccines could lead to epigenetic memory and trained responses?
Well, I think this is a plastic process. So you gain some markers of open chromatin, but you encounter many things in life, and you can thereafter also lose it. That's my feeling. I think it's just a serendipitous process at the moment that we encounter different things during our lifetime, and that's why I think these effects are slowly dying out. Basically, this is in. The changes, the epigenetic changes in the bone marrow progenitors are very different from those that occur in, let's say, an end stage differentiation cells like a T or B memory cells. Even a macrophage is much more rigid, let's say, in the way in which the functional program can change. So I think this is the reason why we change during time, and the different things that we encounter in life shift, and we lose this protection. I think that also now we are in a very special situation, of course, but let's say before the COVID-19 situation, I would have envisaged that only some groups of risk would need such a vaccination, such as very young children, or the elderly who are more susceptible to infections, because in the young adults, actually, the numbers that we would need to vaccinate to protect somebody would have been much lower. Of course, now in the COVID-19, the situation changes dramatically, and even this partial protection for 2 or 3 years can prove to be very beneficial.
Great, thank you very much. There's hundreds more questions, as well as invitations to collaborate with you from members of the audience. Let me see. Let me pick a few more to finish. There's one here. Would you agree that what you've shown us today supports the importance of the development of live attenuated vaccines instead of killed or subunit vaccines that only protect against one pathogen and not that well either? So is it better to go for your approach?
Well, it is not my approach. It was an approach that has been done long time before I was born. So, it is true actually that probably live attenuated vaccines are are more effective and are helpful for more reasons. It is also very tough, however, to recommend one above the others because, of course,
Yes.
there are some side effects. That was the reason to develop the subunit vaccines, the recombinant vaccines, and so on. So there are advantages and disadvantages in both approaches. I do think, though, that we can learn a lot from these effects that are induced by live vaccines to ensure that even the subunit vaccines are adjuvanted properly, that they induce a much better response. Now, in developing countries with high infectious pressure, in which BCG is now given or OPV is given, and it has been shown that they can protect against non-specific infection, in those countries, we have to ask ourselves very critically at the moment that we would like to take away an attenuated vaccine and introduce a subunit vaccine, because in such conditions in which a large number of children are protected through these non-specific effects, it might be deleterious, even if we do have a little bit less side effects, but then we lose a lot of children due to these non-specific infections which are no longer protected. We have to look at the general picture and be very careful that when we take away OPV, for example, now it's an entire discussion. If OPV is taken from the population, we might lose a lot of protection against other infections induced by OPV. So there we have to do studies and study what is happening, and then take the decision based on a broad set of data, not only the data based on the target disease. I think that overall morbidity and mortality would be very important to consider when looking at vaccination programs.
Great. So the final question is about asymptomatic people and why you think that they're able to modulate COVID-19. How do you think that the mechanism of BCG increases recognition of the virus in this group by TLRs, IL-1, IL-6? How do they avoid COVID-19 replication? Yeah.
So we should do experiments and studies on that. We don't know yet for SARS-CoV-2. But based on information on other viruses and other infections, I would think that there is a combination of increased receptor recognition. We know that several receptors are upregulated due to BCG, among which TLRs. So I would expect that some TLRs, such as TLR3, 7, 8, are upregulated and important for the virus recognition. On the other hand, also activation of some of the antiviral mechanisms, including IL-1, IL-6 pathway, and possibly also the interferon pathway, which is obviously very important in antiviral mechanisms, would be responsible for these. And the combination of these factors would lead to an easier elimination of the virus.
Okay, great. So thank you very much, Mihai.
Thank you.
I gather nobody heard my introduction, like you mentioned. Maybe I'll just finish by saying that you are the recipient of the Spinoza Prize in 2016, and you have an ERC Advanced Grant won in 2019. And it was obvious because the presentation was so good. There's so much, so many good comments. People have really, really appreciated the lecture. So we thank you very much. And
thank you.
We look forward to the results of the trial. Thank you so much.
Thank you very much.
Thank you. And thanks to all our attendees for joining, and see you at the next webinar. Thank you.